Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speakers your questions. Your questions in the question and answer window will only be visible to myself and our speakers. So thank you for joining us today. It is now my pleasure to introduce our speakers, Kathleen Kenny and Chris Manzari from WuXi Advanced Therapies.
Hello, I'm Kathleen Kenny, the Associate Director of Viral Clearance. I also have Chris Manzari, the Manager of Viral Clearance Downstream Purification and Virus Production, here with me. Today we are going to provide a high-level overview of viral clearance from WuXi ATU's perspective. There is a risk of viral contamination to all biotechnology products developed from cell lines. Several regulatory guidances recommend a risk-based approach to assess safety. This is done through the three main approaches shown here. The first is through material sourcing by careful selection of raw materials, replacing what you can with non-human or animal-derived products, or additional steps such as pre-treatment of your materials. Next, we have testing for the presence of adventitious agents, such as through several of the assays offered at WuXi ATU's testing services. Then, what we'll be discussing today is viral clearance. VC studies are used to demonstrate actual removal or inactivation of known potential viral contaminants using relevant viruses, and additional viruses with varying biochemical and biophysical properties using model viruses. So what is viral clearance? Stated simply, viral clearance is the validated removal or inactivation of viruses in a given manufacturing process. But in reality, it is much more complex. Viral clearance studies fall into two categories for execution. The first is products and devices which are made by or derived from living sources, or non-biologically derived medical implants. Items from this category include products such as recombinant proteins, cell or gene therapies, and some medical devices. The second category is studies performed to demonstrate the effectiveness of a cleaning process on a specific piece of equipment or manufacturing suite. We perform viral clearance studies because there are many potential sources of endogenous viral contaminants. These come from making use of cells or human plasma to make the target product, which is part of the upstream process. When using cell lines, there are unwanted side products. Bacteria can produce over 100,000 known protein species, while mammalian cells produce over 1 million known species. With products produced in cell culture, there is a significant need for product cleanup, and that is done through the downstream purification process. So now I'll turn it over to Chris to go through considerations for the design of viral clearance studies.
Good afternoon. My name is Chris Manzari. I am the Manager for Viral Clearance Downstream Purification and Virus Production here at WuXi. So just to go over the manufacturing process and following up on what Kathleen had said. For biologically derived products such as monoclonal antibodies and cell and gene therapy products, we like to designate two separate regions of interest, the upstream and the downstream. As far as the upstream is concerned, that is generally the cell culture and bulk human plasma, followed by a clarification to remove any cell debris, at which point we move into the downstream process. So the purpose of the purification process is to have multiple orthogonal means to separate and purify product from other cellular and growth media elements, as well as process-related impurities, as well as product-related impurities. As you can see here, the downstream can be broken down into several distinct steps, including capture, either through affinity or some other means, chemical inactivation by low pH or solvent detergent, as well as polishing columns, and then finally by a nanofiltration through a virus removal filter. So some steps are more robust and some steps are not quite so robust. So, what do we say when we mean robust? Robust is defined in the Q5A regulations as being greater than or equal to 4 logs of clearance or inactivation. We see the more robust steps being in the low pH solvent and detergent inactivations through anion exchange chromatography and through virus removal filters. Now, these are by no means exclusive. We do sometimes see very good at times total clearance through cation exchange or mixed mode. But as a general rule, these 3 designated steps are where you're going to find your most robust clearance. So, getting into the gene therapy, since it is a fairly new form of therapeutic, the virus risk is considered the same, if not greater, when compared to the recombinant space. And that is mainly due to the use, in some instances, of helper viruses. So, WuXi Advanced Therapies currently uses the ICH-Q5A guidance for now. The draft revision of the version 1, which will be revision 2, is currently expected sometime this year. We do expect the regulatory bodies to be reasonable in the application of previous VC requirements to the gene therapy space. Whereas if a viral clearance cannot be reasonably performed on a given virus or a step, it shouldn't be expected. For example, nanofiltration with parvovirus. If there is a helper virus used in the transfection process, we'd evaluate that virus at a bare minimum. For example, baculovirus in the SF9 expression system. And if there is residual helper virus present in a given process step, such as a pre-capture heat inactivation or detergent inactivation, we do work with clients to gain high safety factors to demonstrate a greater than 20 cumulative log reduction factor. For some examples of gene therapy systems, the Vero system that uses herpes simplex or HSV. HSV is required to be evaluated as it is the relevant helper virus. We do look at MVM as a model parvovirus, as well as Rio3 and XMULV, which are, in addition to being analyzed in the gene therapy space, they are also standard viruses in a MAB evaluation. Some unit of operations that we do evaluate, again, are detergent inactivation, where we can see greater than 6 logs of reduction. Affinity capture column, where we see anywhere from 2 to 4 logs of reduction. And 1 or many polishing columns, each with 2 to greater than 6 logs of reduction. And we also do retrovirus grade nanofiltration without the parvovirus, where we see greater than 6 logs of reduction almost across the board. Again, these are estimates. Your results may vary, but based on prior studies that have been done, these are some of the values that can expect to be seen. So we do have a couple of special considerations. For example, with helper viruses, sometimes they are naturally present early on in the downstream process, again, for your affinity column or for your pre-affinity inactivation step. In those instances, we may not need to spike in the HSV. What we would do is we would do a preliminary titer during our toxin interference testing to determine if we do need to add in any HSV to the process step, or if we can use the virus that is native to the sample to determine your inactivation or removal log reduction value. So that is something that we would build into the client protocol when we develop the study process with our clients. So the hows and measurement standards for viral clearance, some of the expectations that are general across the board. For phase 1 and phase 2, generally we would look at 2 viruses. For example, in a CHO-based platform for monoclonal antibodies, we would look at a retrovirus and parvovirus, and those most standard are some form of MULV and MVM. We would look at 1 or 2 column process steps, 1 virus retentive filtration step, and 1 inactivation step. So for this entire process for a phase 1 or phase 2 study, you would need to demonstrate at least one robust parvovirus step, again, with greater than 4 logs of reduction. And the cumulative orthogonal clearance with a safety factor for retrovirus is what you are looking for here. When we move into phase 3 and beyond, the studies get a little larger and a little more complex. We start to look at a 4-virus panel that encompasses a range of large viruses and small viruses, DNA and RNA, as well as enveloped and non-enveloped. And here is where we would consider product origin and other potential contaminants to select a virus panel that would best match what your product is, where it comes from, and what your specific impurities would be for that process. There would be multiple columns with virus reduction filtration, viral inactivation. We would also take a look at new and aged column media, as well as doing carryover for the cleaning and sanitization of your chromatography columns. We also do assess the mass balance by testing additional fractions. For your phase 1 and phase 2, generally we're only going to be looking at your product containing fraction for chromatography. For your phase 3 studies, you're going to be looking at most of your fractions for washes, strip samples, sanitization, etc. And these studies are also much larger, and they do take a decent amount of time longer to not only develop the protocol, but as well to execute the actual spiking studies. So what is the LRV that we discuss? So the log virus, we take it into account that the log of the virus that goes into the system minus the virus or the log of the virus that comes out of the process. And this is generally going to be either your product, your elution from a column step if it's bind elute, or your flow through if it's a flow through column, as well as your post inactivation and post filtration samples. So it's really going to be your log 10 in minus your log 10 out. For example, if you put in 7 and you get out 3, your log reduction value is going to be 4. So the per Q5A from ICH revision 1. So the entire purification process should be able to eliminate substantially more virus than is estimated to be present in a single dose equivalent of your unprocessed bulk. Kathleen, back to you.
All right. Here at WuXi, we have developed a fine-tuned approach to the virus spiking strategy. Instead of the more typical percent spike approach shown on the left, we will spike targeting a specific viral load. With this, we spike virus at the right concentration, where the virus titer is considered so as to not overload the system, but is still a sufficient amount of virus per regulatory guidance. On the right, you can see the increased LRV with WuXi's targeted viral load and large volume testing approach for each step. This increase per step could help clients reach their desired cumulative LRV with fewer steps overall. So in order to obtain the titer for each of your samples and calculate the LRV, we'll use one of our key assays. One of the assays we use here is qPCR. So with qPCR, we are measuring the amount of specific virus DNA or RNA in a given sample. The output is given in genomic copies. qPCR is highly specific and highly quantitative. However, qPCR is measuring any DNA or RNA of the virus in a given sample, so it could overestimate the live infectious virus. We also utilize the TCID50 assay. This assay is performed in 96 well plates. The assay is scored based on a percentage of cell death, with wells scored as either positive or negative for viral infection. However, limitations to sensitivity occur at lower concentrations of infected wells of this assay. Alternative calculations are then needed when only a few wells of a critical sample are infected. Additionally, it is more difficult to increase the TCID50 assay for large volume testing. So WuXi's preferred method, when possible, is the plaque assay. It has higher sensitivity than other infectivity assays. It'll measure the relevant infectivity of a given virus within a sample. By placing a series of dilutions of a virus-containing sample on the monolayer cells, plaques form, which are then counted to obtain the titer. Most importantly, it is much easier to scale up to test larger volumes. Utilizing the power of the plaque assay, large volume testing for steps where we expect full clearance leads to increased log reduction values. We can see the benefits of large volume testing in this slide. The first volume of the filtrate sample shown is the standard volume tested in the plaque assay. When about 8 logs of virus is in the load sample, we may see 5.3 logs of virus reduction with the standard 1.5 mils tested. When you increase the volume tested, the amount of log reduction will also increase, assuming there's little to no virus. When 21 mils of samples tested, we could achieve almost 6.5 logs of clearance. And with 70 mils, we may see greater than 7 logs of clearance. This is a simple to execute change in the lab, which can add greater than 1.5 logs of virus reduction for a single step. Large volume testing for product-containing samples could allow a client to evaluate less process steps overall, which then would reduce the study cost and time. Overall, WuXi ATU offers many features that lead to better study design from the beginning. The targeted viral challenge improves the total LRV you can receive from each step. The plaque assay allows for large volume testing, which can increase your total LRV. With over 25 years of conducting viral clearance and a database with over 3,500 studies, we can perform data mining to appropriately design your study. Lastly, WuXi viruses are produced in-house. The purification techniques result in high quality and high titer stocks. And in addition to the assays and resources available, we have a highly trained technical expert staff. Our team is comprised of 1 director, 3 managers, 3 supervisors, 6 scientists, and 25 technicians on multiple shifts. The validation on demand group supports clients who cannot travel to the site for study execution. This team has experience with multiple column and resin types, as well as experience performing filtration steps using devices from several vendors. The validation on demand team generally performs 9 to 11 studies per year, and these studies have higher collaboration with the client in order to appropriately design the test methods, perform the technical transfer runs, and complete the final review of the test methods for execution. So that concludes our presentation for today.
So the first question that we have is, are there any disadvantages to doing the percent spike method over the targeted spike approach that WuXi ATU employs?
Sure. So virus titers can vary from lot to lot, and improvements in purification are leading to higher starting titers. When using the percent spike approach, only the volume is really taken into consideration. This means for 2 studies with 2 different lots of virus, you may use the same percent spike, let's say for a filtration step, but then you end up adding 7.5 logs of virus in one and 8.5 logs of virus in the other. This could lead to a couple of issues. The first is that the filter with 8.5 logs of viral load could be over-challenged and therefore not achieve complete clearance. Additionally, the potential LRV for the filtration step is dependent on the total viral load at the start. Even if you achieve complete clearance in both of these examples, you may see a log or higher difference in the results. If this difference is occurring between similar molecules or between your phase 1 and phase 3 studies, you may need to provide additional justification when filing. So using our target viral load approach, you could always target 8 logs of virus in all the runs you perform. This gives you more consistent results as it's one less variable to consider. Additionally, knowing the starting viral load allows for strategic pre-planning of the large volume testing in order to achieve your maximum potential LRV.
Okay, and you mentioned that nanofiltration for parvovirus and some gene therapies wouldn't be performed. Can you explain why that is?
Excellent question. So the reason parvovirus isn't evaluated for nanofiltration is because the filters that are designed specifically for parvovirus filtration for monoclonal antibodies generally have a nominal pore size of 20 nanometers. Unfortunately, a lot of AAV products fall into that 20 nanometer range. So if you were to run a parvovirus filter to evaluate your AAV product, you would not only be retaining your parvovirus that is spiked into the sample, you would also be retaining the AAV. And that is why for a lot of the gene therapies that are adeno-associated viruses, they have a nominal pore size of 35 nanometers. And that is a pore size which not only the AAV would go through, but also the parvovirus.
And what are the advantages and disadvantages of performing a study on site versus remote execution?
Another excellent question. So again, there are, like you said, advantages and disadvantages. Some of the advantages to performing a remote study or validation on demand is one that you don't have to actually come to our site and spend a week, 2 weeks, 3 weeks, depending on the length of your study. Living in a hotel, eating out at restaurants. As well as being on site, executing the study yourself, 4 days a week, 5 days a week. There is also when your scientists are on site executing a study, they are not actually in their development lab working on the next, the next product coming down the line. As far as the advantages, it is a very collaborative process. So it is all of the documents that are done, except for research and development studies, are QA reviewed internally. They are also fully auditable by regulatory authorities as well as our clients. We also have, again, like Kathleen had said, we have a very, very well trained, very highly talented team that is able to execute on your behalf. Again, it is also very collaborative. So the clients are involved in all phases of the document drafting from the initial draft, which the clients will review. There is also a lot of back and forth in there. The technical transfer process is something that that is built into our process, where we will execute every process step. So that would be every column and every nanofiltration step that will have virus introduced to it. But for the technical transfer, we do that without the introduction of virus. This is to demonstrate that the validation on demand team can perform the scale down such that it is virtually identical to anything that the client has done on their bench, and it matches their manufacturing process as far as chromatograms, as far as product yield and product concentration in whatever fractions are being analyzed. We do not proceed with the spiking steps until all the client is satisfied that the technical transfer has been performed to their satisfaction. And not only that, it is, in reality, it's a time saver for the client, even though for validation on demand studies, the actual overall execution of the study, because of the tech transfer, is generally 2 weeks longer than the timeline for a regular study. But as long as the studies are planned out, we usually can accommodate just about anything.
Okay, great. And what effect would high buffer cytotoxicity, sorry, let me see this again. What effect would high buffer cytotoxicity have on potential LRV?
Sure, I can take that one. So the sensitivity of the assay depends on the volume of the fraction and the dilution of the sample. If a fraction has high cytotoxicity, the sensitivity of the assay is reduced. This is factored into the overall titer calculations and can decrease the potential LRV for that given step. So if we are looking at very high cytotoxicity, there are a few changes that we can incorporate to help overcome the effect of this dilution. The first is simply test more volume of the fraction. As shown in a previous slides, increased volume can lead to increased LRV because we're more likely to detect any virus that is still in the sample. The second change we can make is to increase the spike target viral load. Both of these options really are only beneficial when we can expect complete or almost complete viral clearance in the step.
Okay. And what is the LOD and LOQ for the plaque assays?
We define the limit of detection of the plaque assay as 1 plaque because we really can't detect less than 1. The LOQ is the lowest level that an analyte can be quantitated with any degree of certainty. In viral clearance studies, this is really governed by the ICH Q5A guidance, and the Poisson distribution is applied to determine the maximum theoretical amount of virus present in each sample with 95% certainty. In the plaque assay, the LOQ is going to vary for each sample since it's based on the Poisson distribution, the amount of inoculum plated per well or large volume dish, the number of wells or dishes tested, and the dilution of the sample.
And are some detergents more effective than others? If so, can you give an example?
Sure. So we don't determine which detergents are used for a viral clearance study, but we can help in giving some data prior to the start of a study. We've seen numerous different detergents at a wide range of concentrations over the last several years of viral clearance studies. I'd say the most common we see is Triton X100, but studies have been completed with Tween, LDAO, SDS, and more. Generally, we're going to find complete or almost complete clearance for envelope viruses with most of these detergents. However, I really can't go too far into determining which one would be the most effective without knowing the final concentration of that detergent, the processing times and temperature, and the non-toxic dilution. If a client did have some of these factors known during the design of the studies, we could certainly use WuXi's viral clearance database to give further guidance and help define the concentration or the direction they want to go with their detergent.
Okay, well, thank you, Kathleen and Chris. And thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website. And as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.